Hello students, how is everything? I hope everything is well with you. I'm back with a new tutorial video on plant breeding chapter 4 reproduction in crop plant and its relation to breeding methods. I want you to be with me until the end of this video and don't forget to hit the subscription button and share the channel. Let's start the content with asking ourselves what is the importance of knowing reproduction methods as well as pollination type in plant breeding. You see, plant breeding is science of improvement. Thus, if you want to understand breeding methods which will be discussed starting from Chapter 5, first you should have basic understanding about the foundations. That's why it is recommended to have basic understanding about selection types, germplasm, and crop domestication, and evolution. When we came to the previous question we raised, there are 4 main reasons why we should know mode of reproduction or pollination. These are 1. To select appropriate breeding method because the methods of breeding depends upon the mode of reproduction. 2. To understand effect of outbreeding and inbreeding as this situation depend on genetic structure of a given crop. We will discuss what inbreeding and its effects on coming slides. 3. To determine genetic constituent/structure of plant and population. Self-pollination promote homozygosity, while cross-pollination promote heterozygosity. 4. For handling and management of breeding program. This situation related to maintenance. For example, the maintenance period for hybrid variety, synthetic, and pure line is not the same. I want you all to remember the above-mentioned reasons. When we came to crop or plant reproduction methods, we can classify reproduction methods in plants into 2. These are sexual and asexual reproduction methods. Let's see what makes this group different from each other and their advantages. Reproduction refers to the process by which living organisms give rise to the offspring of similar kind. Species. 1. Asexual reproduction refers to multiplication of plants without the fusion of male and female gametes. Asexual reproduction can occur either by vegetative plant parts or by vegetative embryos which develop without sexual fusion or apomixis. Therefore, asexual reproduction is of 2 types. The first one is vegetative reproduction and the second one is by apomixis. Apomixis refers to the development of seed without sexual fusion or fertilization. In apomixis, embryo develops without fertilization. Thus, apomixis is an asexual means of reproduction. Apomixis is found in many crop species. Reproduction in some species occurs only by apomixis. This apomixis is termed as obligate apomixis. But in some species, sexual reproduction also occurs in addition to apomixis. Such apomixis is known as facultative apomixis. Vegetative reproduction refers to multiplication of plants by means of various vegetative plant parts. Vegetative reproduction is again of 2 types. The first one is natural vegetative reproduction and the second one is artificial vegetative reproduction. 2. Sexual reproduction. Sexual reproduction involves fusion of male and female gametes to form a zygote, which develops into an embryo. In crop plants, male and female gametes are produced in specialized structures known as flowers. Thus, understand flower structure is good to understand mode of pollination in crop plants. So let's discuss basic points regarding flowers. A flower usually consists of sepals, petals, or their modifications, stamens, and or pistil. A flower containing both stamens and pistil is a perfect or hermaphrodite flower. If it contains stamens, but not pistil, it is known as staminate, while a pistillate flower contains pistil, but not stamens. Staminate and pistillate flowers occur on the same plant in a monoecious species, such as maize, colocasia, castor, coconut, etc. But in dioecious species, staminate and pistillate flowers occur on different plants. Example: papaya, date palm, pistachio, etc. The other important topic to discuss in this section is what is the significance of sexual reproduction. Sexual reproduction makes it possible to combine genes from 2 parents into a single hybrid plant. Recombination of these genes produces a large number of genotypes. This is an essential step in creating variation through hybridization. Almost the entire plant breeding is based on sexual reproduction. Even in a sexually reproducing species, sexual reproduction if it occurs, is used to. Advantage. Example. In sugarcane, potato, sweet potato, etc. The other important point to discuss in this topic will be about anthesis and modes of pollination or inbreeding and outbreeding mechanisms. What is anthesis and why it's important? Anthesis is the first opening of the flower in the process of flowering. It is greatly affected by the environmental factors like humidity and temperature. Knowledge of anthesis is desirable to make successful crosses, and it largely determines the mode of pollination. Pollination is the process by which pollen grains are transferred from anthers to stigma is referred as pollination. Pollination is of two types. The first one is autogamy, or self-pollination, and the second one is allogamy, or cross-pollination. Let's discuss them one by one. 1. Autogamy is a type of pollination when transfer of pollen grains from the anther to the stigma of same flower is known as autogamy or self-pollination. Autogamy is the closest form of inbreeding. Autogamy leads to homozygosity. Such species develop homozygous balance and do not exhibit significant inbreeding depression. Mechanism promoting self-pollination. 1. Bisexuality, which is presence of male and female organs in the same flower, is known as bisexuality. The presence of bisexual flowers is a must for self-pollination. All the self-pollinated plants have hermaphrodite flowers. 2. Homogamy, which means maturation of anthers and stigma of a flower at the same time. As a rule, homogamy is essential for self-pollination. 3. Cleistogamy is a situation when pollination and fertilization occur in unopened flower bud. It is known as cleistogamy. It ensures self-pollination and prevents cross-pollination. Cleistogamy has been reported in some varieties of wheat, barley, oats, and several other grass species. 4. Chasmogamy. Opening of flowers only after the completion of pollination is known as chasmogamy. This also promotes self-pollination and is found in crops like wheat, barley, rice, and oats. 5. Position of anthers. In some species, stigmas are surrounded by anthers in such a way that self-pollination is ensured. Such situation is found in tomato and brinjal. In some legumes, the stamens and stigma are enclosed by the petals in such a way that self-pollination is ensured. Examples are green gram, black gram, soybean, chickpea, and pea. Genetic consequences of self-pollination. Self-pollination leads to a very rapid increase in homozygosity. Therefore, populations of self-pollinated species are highly homozygous. Self-pollinated species do not show inbreeding depression, but may exhibit considerable heterosis. Therefore, the aim of breeding methods generally is to develop homozygous varieties. 2. Allogamy or cross-pollination. Allogamy is a type of pollination where transfer of pollen grains from the anther of one plant to the stigma of another plant. This is the common form of outbreeding. Allogamy leads to heterozygosity. Such species develop heterozygous balance and exhibit significant inbreeding depression on selfing. Mechanism promoting cross-pollination. 1. Dicliny. It refers to unisexual flowers. This is of two types. The first one monoecious and the second one is dioecious. When male and female flowers are separate but present in the same plants, it is known as monoecious. In some crops, the male and female flowers are present in the same inflorescence, such as in mango, castor, and banana. In some cases, they are on separate inflorescence, as in maize. Other examples are cucurbits, grapes, strawberry, cassava, and rubber. When staminate and pistillate flowers are present on different plants, it is called dioecious. It includes papaya, date palm, spinach, hemp, and asparagus. 2. Dichogamy. The term dichotomy came from the Greek diko, apart, and gamous, marriage. It refers to maturation of anthers and stigma of the same flowers at different times. Dichogamy promotes cross-pollination even in the hermaphrodite species. Dichogamy is of two types. The first one is protogeny and the second one is protandry. When pistil matures before anthers, it is called protogeny, such as in pearl millet. When anthers mature before pistil, it is known as protandry. It is found in maize, sugar beet, and several other species. 3. Heterostyly. When styles and filaments in a flower are of different lengths, it is called heterostyly. It promotes cross-pollination, such as linseed. 4. Hercogamy. Hindrance to self-pollination due to some physical barriers, such as presence of hyaline. Membrane around the anther is known as hercogamy. Such membrane does not allow the dehiscence of pollen and prevents self-pollination, such as in alfalfa. 5. Self-incompatibility. The inability of fertile pollens to fertilize the same flower is referred to as self-incompatibility. It prevents self-pollination and promotes cross-pollination. Self-incompatibility is found in several crop species like brassica, radish, nicotiana, and many grass species. It is of two types, sporophytic and gametophytic. 6. Male sterility. In some species, the pollen grains are not functional. Such condition is known as male sterility. It prevents self-pollination and promotes cross-pollination. It is of three types. First one is genetic, the second one cytoplasmic, and the third one is cytoplasmic genetic. It is a useful tool in hybrid seed production. Study of floral biology and aforesaid mechanisms is essential for determining the mode of pollination of various crop species. Moreover, if selfing has adverse effects on seed setting and general vigor, it indicates that the species is cross-pollinated. If selfing does not have any adverse effect on these characters, it suggests that the species is self-pollinated. The percentage of cross-pollination can be determined by growing a seed mixture of two different varieties together. The two varieties should have marker characters, say green and pigmented plants. The seeds are harvested from the recessive, green, variety and grown next year in separate field. The proportion of pigmented plants in green variety will indicate the percentage of outcrossing or cross-pollination. Genetic consequences of cross-pollination. Cross-pollination preserves and promotes heterozygosity in a population. Cross-pollinated species are highly heterozygous and show mild to severe inbreeding depression and a considerable amount of heterosis. The breeding methods in such species aim at improving the crop species without reducing heterozygosity to an appreciable degree. Usually, hybrid or synthetic varieties are the aim of breeder wherever the seed production of such varieties is economically feasible. These are the main points to be discussed in Chapter 4, and I hope you get so many important concepts from it. But before concluding today's video, I want you to remember one last point, that is the importance of pollination. The mode of pollination plays an important role in plant breeding. It has impact on 5 important aspects. 1. Gene action. 2. Genetic constitution. 3. Adaptability. 4. Genetic purity. And 5. Transfer of genes. The other remaining topic in this chapter will be about determination of mode of pollination and natural outbreeding. Therefore, let's talk about how can we determine a given crop whether self or cross-pollinator. Generally, 3 ways. The first one is by critically examine its flower, and the second one is by isolating single plant and observes whether or not seeds are produced. Isolation can be in space, in time, or isolation by bags or cages. Inbreeding effect is one good indicator of self-pollination. For natural outcross determination, there are procedures, and these are: 1. Interplanting varieties carrying dominant and recessive genes. Then, seed is harvested from the recessive gene, and then the seed is planted and the number of dominant plant present in the population is counted, and their amount could indicate the amount of the cross-pollination. This all for today's video, and I will back with other interesting content. Until then, I wish you all peace.